Hello, everyone. I'm Lu, a postdoctor from the Division of Vascular Surgery from Dr. Sita Lab. And today I'm really appreciate this opportunity to share our current work about vascular Ehlers-Danlos syndrome by using patient-derived iPSC-based vascular models. To begin this, I would like to give us more introduction about the Ehlers-Danlos syndrome. Yes, EDS is a genetically heterogeneous group of inherited connective tissue disorder. So we can see in the figure, the patient present the joint hypermobility and hyperextensible skins. So currently, it characterized into 13 subtypes. So among these 13 subtypes, the vascular EDS is the most severe subtype called VAS. And vEDS is a rare connective tissue disorder. So this rare vascular disease is mainly caused by the mutation at the collagen 3A1, this gene, called the defect in type 3 collagen. And in the patient, it affects the blood vessels more. So the patient will prompt to develop aneurysm, which is dilation of the part of the some blood vessel, and aortic dissection, which is an intimal tear and lead to the blood leaking into the surrounding layers. So the most affected cell types is endothelial cells and the smooth muscle cells. So at present, what we all know about the VAS, it came from the VAS mouse model, where this mouse model carried the pathogenic collagen 3 mutations. And this mouse model also recaptured the key features of the VAS, for example, the dermal and vascular fragility, the aortic rupture, and when study point the signaling of PLC, PKCRK, this signaling is dysregulated in this model. And also several drugs, including the angiotensin II receptor blocker and celiprolol, was showed the potential therapeutic in this mouse model. And as we may all know, that during the aneurysm formation, the endothelial dysfunction play a really key role in this. So this draws us as a really key question is that how do this collagen A3 mutation drive endothelial dysfunction and lead to this arterial pathology? So to answer this question, first we want to address what are the endothelial phenotypic abnormalities associated with VAS? So to answer this question, I want to introduce first a very powerful tool of induced pluripotent stem cells, iPSC cells. This discovered by Yamanaka, and these cells just need a small amount of the blood and reprogramming to iPSC cells, and then we can differentiate these iPSC cells into the endothelial cells and the beating cardiomyocyte, as we can see here. And these cells can carry the genetic background from the patients, which is a really powerful tool to study rare vascular diseases. So the Bozuki Biobank within the division of vascular surgery, generating the iPSC cells from the patients with vascular diseases. So through our biobank, we recruited 5 VAS patients, 4 the mutation at the gene collagen 3A1 and 1 at collagen 12A1. And we reprogramming all these cells to iPSC cells. And then we differentiation to the endothelial cells. So as we can see here, compared to healthy control, the iPSC-EC from VAS patients showed abnormal morphology. And then when we want to characterize the function of these cells, we observed that the iPSC-EC from VAS patients showed the decreased capacity of angiogenesis, LDL uptake, and the nitric oxide production. So it means that this iPSC-EC exhibits the impaired endothelial function. So to further detect and study the underlying mechanism, we performed the bulk RNA sequencing from the iPSC cells from 3 VAS patients and healthy control. We found that the surprise gene is highly associated with one signature is regulation of vascular permeability. It means that this loss of the barrier control, it means that it's increased permeability. So to functional validate this, we performed a vascular permeability assay. We found that in the iPSC-EC from the VAS patients all showed the increased permeability, which is consistent with our transcriptomic result. We also found the significant upregulation of MMP2 MMP2 and MP9, these two genes. And we know that compared to the healthy arterial, for the aneurysm part, the MMP2 can produced by the dysfunctional endothelium, and this can like induce the exocyte matrix degradation and promote the aneurysm formation. So based on what we found here, we want to address the second question. How does coding mutation in vEDS patients lead to the endothelial dysfunction? So to answer this question, we performed the parameter editing to correct the pathogenic allele in the patient-derived iPSC cells, so which showed it's consistent with the healthy control. And then in the differentiated endothelial cells, we found that compared to the patient-derived IEC, the pathogenic correction can reverse this capacity of the angiogenesis, LDL uptake, and nitric oxide production. So it means that this correction can restore the EC dysfunction, what we found in the vEDS patient. And highlightedly, we've also found this pathogenic correction can normalize the endothelial permeability, bring the level back to comparable to the healthy control. And based on the transcriptomic result, we found this pathogenic correction can reinstate the cardiac and muscle signatures. As expected, we found that also the upregulation of MMP2 and MP9 was also decreased in this pathogenic correction, which is corresponded to what we found before. So for now, we found that is all in vitro work. So we want to know what happened in vivo. So through the division of the vascular surgery, we obtained the arterial tissue from the vEDS patient and performed the single cell multi-omics analysis. And our preliminary data showed there's a clear cluster for the endothelia. And we will validate our key findings in follow-up studies. So in summary, we found the vEDS iPSC-derived endothelial cells exhibit impaired endothelial function and increased the vascular permeability linked to upregulation of two genes, MMP2 and MP9, and this can be reversed by the pathogenic correction. So thanks for your attention, and I'm grateful for all our lab members and collaborators to make this work possible, and I'm happy to take questions. Thank you.